This is CGTN, China Global Television Network.
Hello and welcome to this Razor COVID-19 special. I'm Emma Keeling. Actually, while most of the show is about the virus, first up we thought you might enjoy a little bit of a break. The pandemic has meant much of the scientific research going on around the world has ground to a halt, and other researchers have switched their focus to studying the new virus. However, some research projects have managed to go ahead despite the pandemic by finding new ways to work.
The Nippon Foundation GEBCO Seabed 2030 project has the ambitious goal of mapping the world's oceans by 2030. The collaboration involves teams from all over the planet. Professor Rob Beeman and his team are mapping the seafloor around Australia.
If you compare it against Mars or the Moon, now both Mars and the Moon are 100% mapped from satellites that we've sent out. 100% of those planetary objects is mapped. But here on the Earth, only 20% of the world's oceans have been mapped. That means there's an awful lot yet to discover. Now here is a classic example, the Coral Sea Marine Park. It's just offshore, it's not far from where I live. And yet, we only really knew, I guess, the detailed shape of the shallow portions of it. The deeper parts of it were a bit of a mystery. And so this voyage has turned this, I call it like a blank canvas. You know, if you're thinking of like a painter's blank canvas, that was kind of the picture that we had. But now we've really filled in that picture.
The team maps the seafloor using multi-beam sonar, which sends out sound pulses or pings, which bounce off the seafloor and return to the ship. The system calculates the time each pulse takes to return and translates that data into a 3D image.
So as the ship's going along, you build up almost like a three-dimensional carpet of soundings, depth soundings. It's quite detailed, and so you end up with this beautiful three-dimensional shape of the seafloor. As long as the ship is moving forward, that picture just builds up and up. The coral has managed to keep growing. Ultimately, you end up with something that's nearly 1 kilometre high. But because parts of it have actually collapsed, we've been able to look in, like an X-ray layer, looking at all these rock layers built up through time. And so it's quite exciting to put the camera there and fly it, you know, up that 1 kilometre high coral reef.
As well as mapping the seafloor, they also send down a remotely operated vehicle, or ROV, known as Sebastian, with other images in mind.
It's a remote operated vehicle. It's tethered to the surface, so it actually has a cable running up to the surface, a fibre optic cable. It has up to 10 cameras. But the beauty of having the 10 cameras is, I think they had 27 monitors inside the ROV operations room. So they had this beautiful, almost 360-degree picture of what was happening around the ROV, and then they could drive it to where it was going. And the vision was so good, I felt like I was standing on the seafloor just watching the seascape drifting past, and, you know, whatever animals. I just never got tired of it. I was always astounded. Even in places that you thought that might be, you know, I wouldn't say boring, but there was just things that, you know, it might be just sandy seafloor. You know, there might not be a lot of obvious animal life. And then literally a minute later, something would drift into view. So there was just this constant surprise, you know. I'd have just enough time to rush out, grab a cup of tea, come back, and there'd be something amazing drifting into view. A good example of that was, I think it might have been the last dive. There was a Dumbo octopus came into view, because a Dumbo octopus is an amazing animal. You know, it's got these beautiful big flappy ear things, and they could see it on one of the other cameras. And so, you know, the whole camera was like tilting this way, and we just got this beautiful picture of the Dumbo octopus coming into view. And then we just followed it down.
The voyage was planned to commence just as COVID-19 travel restrictions were put in place, which meant that scientists who would normally be aboard had to come up with a remote system.
So we had to figure a way on how to, manage from shore and be an integral part of a research expedition with no scientists on board. So we had, the technical challenges of being able to communicate with the ship. We also had software that allowed us to look at monitors on board the ship so we could actually see what was being collected in real time.
The steep coral atolls provide a habitat for a rich array of life, even at depths of up to 1 kilometre.
We're talking temperatures around 4 to 5 degrees Celsius. Pitch black, it's deep, it's dark, it's cold. And I guess that was surprising too, just how much marine life was down there.
How were you able to actually see things at that depth?
You need light. It is completely pitch black down there. You needed to turn on these very strong strobes. And of course there was lots of colour there. All the animals had lots of different colours.
It kind of makes you question why nature has created those vibrant colours when there's no light down there.
There seems to be a lot of animals that are coloured red in these deeper oceans. You know, there are deep amphipods, crustaceans, some of those Dumbo octopus, a deep, deep red, almost scarlet, almost purpley red colour. What we do know is red, that wavelength red, is absorbed very, very quickly. One of the really good camouflage colours to use is red. It guarantees that you're going to be completely invisible. But yet there were plenty of other animals down there with lots of coloration. We saw lots of fish, even in these deeper zones. You know, they had colour on them, quite strange colour. There was coffin fish with beautiful yellows and purple colours, and, you know, just strange coloration. Some fish were almost mirror-like, like mirror scales.
Why is it important to have mapped the world's oceans? What are we going to gain from that knowledge?
When you start looking at, you look at the Earth and you understand that 7/10 of that globe is underwater. So straight away we can contribute 35,000 square kilometres of brand new seafloor mapping data to that global effort. From Australia's perspective, we now have, a really good understanding now of the deeper parts, what's living in the deeper parts of the Coral Sea Marine Park. That's of huge benefit to us because, we are the custodians for this part of the world.
The world is still in the first wave of the pandemic, but a surge of new infections has scientists worried that the containment achieved in lockdowns could be rapidly undone. In the UK, there are fears mixed messages from politicians are contributing to a breakout of cases. And America has seen a surge in multiple states, which has some officials worried they may never get the disease under control. Summer has arrived in the Northern Hemisphere. As lockdown restrictions were relaxed in the UK, people took advantage of the warm weather at the beach. But an outbreak in the city of Leicester is worrying Dr. Bharat Pankhania, a former consultant in communicable disease control at Public Health England. Do you feel that the UK eased the lockdown restrictions too soon, too fast?
Oh, without a doubt. I have been saying this from the moment Prime Minister Johnson said we're going to lift the lockdown measures. The number of cases in the community has dropped, which is good, but it hasn't carried on dropping. It has plateaued. Secondly, we are not testing extensively. On top of that, for control measures, you need to be able to identify the cases and their contacts.
Across the Atlantic, American health experts are also concerned that political leaders rushed to reopen. Now states that eased lockdown restrictions earlier are seeing a surge in new SARS-CoV-2 cases. Is America seeing a second wave, or is it still part of the first wave?
This really is still our first wave. So a lot of the reason we're seeing these increasing cases is because we never really fully contained what was going on initially. And so we opened up too quickly. And now we're seeing the after effects of that. We're seeing cases go up. We're seeing healthcare system's becoming overwhelmed again.
Dr. Krutika Kupali is an infectious disease physician who's been helping communities and healthcare officials better prepare for COVID-19. President Donald Trump said cases in the US are going up because we are testing far more than any other country. So how much of the surge can you put down to increased testing?
Oh, I don't think that the increased number of cases are all due to increased testing. Maybe some of it, but really, as we see more people being admitted to the hospital and more people requiring ICU beds, that is definitely not a function of increased testing. That is definitely a function of more cases. And the other thing you have to look at is you have to look at the percentage of cases. And so the percent is going up. And also you have to look at what is going on with testing. So with testing, really, if the outbreak is appropriately controlled, as you continue to test, you should see the numbers actually go down. That's not what we're seeing here. We're seeing, as we continue to test, the numbers go up.
Many American states have paused reopening plans as cases rise. Are we going to see waves, or is it always going to just be this constant surging of cases?
The hope is that we get things down to a point where we can have things like good testing, contact tracing, with the ability to do good isolation and quarantine, so we don't have these surge of cases. The mainstay of any infectious disease outbreak is being able to test, being able to trace, and then being able to effectively isolate and quarantine. And that's really what we need to be able to do in order to have a good, robust response, not just here, but all over the world.
Public health officials in the UK are watching America closely. We've heard officials in America say that they're concerned that if cases keep surging, that they'll never get it under control. What are your thoughts on that?
Yes, I'm very concerned about that, and not only for the United States or the UK, but for poorer nations. And I'm almost convinced, in fact, I'm pretty convinced that the coronavirus, as it is, will become an endemic strain. What I mean by that is that it is persistent. It persistently circulates in our population. And then we will have to get used to living with this virus because it will persistently always be ever present and circulating.
He wasn't surprised to see the virus outbreak in Leicester, but he is concerned how slowly the government was to hand over the data they'd gathered in the area to local officials.
The people in Leicester, the officials, the officers, were unaware that the number of cases in the community had been creeping up and had crept up significantly, and they were blindsided by not knowing this. We should have the data in real time in both places.
Dr. Kupali understands people's need to return to normal, but says the easing of restrictions must be careful and deliberate. She co-authored a paper exploring what the Ebola epidemic taught medical researchers about viral outbreaks, lessons that can be applied to COVID-19.
I think one of the key points that I always tell everybody is that it shows how important it is to invest in global health and research and development. And then I think the other thing that's very valuable would be the importance of community engagement. There was a lot of community resistance in this particular outbreak, and if it hadn't been for the wonderful frontline workers who put themselves at harm's way, working to engage the community and educate them and doing contact tracing, we would not be where we are today, able to say that that outbreak has been declared over. And the cornerstone of that Ebola outbreak has been testing, contact tracing, and education. And I think people really can learn a lot of lessons from that.
After months of lockdown, scientists around the world have noticed pandemic fatigue. And Dr. Pankhania believes this has been compounded in the UK because the government's advice to the public hasn't been clear enough.
I'm very concerned that we will undo the economic cost, the hardship, the pain of the lockdown. This can be undone very, very quickly. It's a very fragile achievement to have brought the case numbers down. It is fragile. And for us to encourage people to say, the mixed message is, go to the pub, enjoy a drink. That's not what I am out for. I'm saying the message should be the virus has not gone out. We're lifting the lockdown. We want you to be measured and careful. Please be careful, even if you're having a drink.
What can we learn from the surges that have happened in communities?
So the science is very clear and it is not difficult to explain, which is if you act fast, if you act early, if you act in a timely manner, you can achieve control.
How will your world change today? What happens here? What happens there? Or what you make happen for yourself? If it matters to you, it matters to us too. Your stories are the stories that need to be told. Africa Live. Find your voice.
Creativity, technology, and inspiration come together in a new series of podcasts from CGTN Europe. What ideas are out there for enriching our common future? Find out in This Way Forward, available wherever you get your podcasts.
Earlier in the year, Raiser's Joe Colan met Professor Chris Tumazu. His company DNA Nudge has developed genetic testing cartridges that help people make healthy food choices based on their DNA profile. But since then, Professor Tumazu and his team have adapted their process to test for the SARS-CoV-2 virus.
DNA Nudge is the brainchild of Imperial University's Regis Engineering Professor Chris Tumazu.
We differ by about 0.1%, and it's that 0.1% that differentiates us from whether we got propensities to genetic diseases, whether or not we can metabolize various foods, whether or not we can metabolize drugs, etc., etc. Okay. This chip here has 96 wells. So effectively, we've taken propensities for things like type 2 diabetes, obesity, hypertension. These are these genetic errors. We then spread those across those wells. So once your DNA is then extracted from your saliva, your DNA is then spread over those wells. And once it's matched to a primer, we know that you've got that genetic propensity. If it doesn't match, you haven't got that propensity. So it's a process of matched and unmatched sequence, basically, on this particular chip. And that's all done in situ in an area as small as this. This replaces an entire laboratory, replaces white coats, pipettes, machines, 3 or 4 weeks of lab time, the cost, the speed, everything is done. And more importantly, privacy. The fact is that because your genetic sample, whether it's a cell or saliva, is actually within the device itself, once the test is done, this is thrown away.
So Chris, when I filmed with you earlier this year, you had just launched the DNA Nudge cartridges, which were designed to help people make better food choices. And since then, you've adapted the cartridges to a very different purpose related to COVID-19. What can you tell us about that?
Back in March, it was actually March that was the eureka moment. You know, we were sitting down in the lab and we started thinking, well, look, you know, all we have to do to this is basically change the primers on our chip, the bait, to bait that can detect effectively corona. That was the first step. And to change some of the mix in the cartridge to transcript from RNA to DNA. So once you've inserted the swab into the cartridge, we're able then to extract the virus, introduce it into our cartridge and do the initial testing. And that's when we went, wow, because it's almost binary. You've either got the virus or you don't. That's the beauty of it, see. Unlike the genetics that we were looking at, where we're looking at mutations in our DNA, in our sequence here, we're just looking at, do you have the viral RNA or don't you?
And you can be asymptomatic and still have the virus.
Exactly, and it can be quite infectious. In fact, one of the clinical virologists at St. Mary's had a very, very small cough on the first day we tested this, and we asked him to go to his car and just test himself with a cartridge, which he did, and he was so positive. He would not have known. He was so positive. One of the strongest signals we'd ever seen. So he went home into isolation. But what we did is so imagine if he hadn't, and he could have spread it, obviously. We gave him 5 cartridges to go home with. So every day, for 7 days, he distributed a swabbing of 5 cartridges. So he stored 5 cartridges. So day 7, he came out and had another swab. And what we were able to do is measure through this, detect the virus on those 5, 6, 7 days. And we saw the regression of the virus until the 7th day. There was a very weak positive signal. The whole idea, has been to reduce the throughput. As we know in the field today, PCR tests, the big testing is taking some 24 to 48 hours. And that's because there's this huge manual process. Once the swab is taken, it could be a nasal swab. I mean, here's a typical swab that's used in the field. A nasal swab that might go into the nasopharyngeal up in the nostrils or in the throat. This then has to go into viral transport media to keep the virus alive. Then it has to go to a laboratory, a very, very high quality, very well contained laboratory. From that, one extracts the RNA, and then that goes into a big PCR machine. And that machine not only does one individual's test, it tests for many individual targets. And then after that, the results get then processed, they then get recorded, and then it goes back to the hospital, then the patient. So with this, we can do it instantly. I mean, the sample bit, the throughput has gone completely down because we'll take a swab, maybe a quick nasal swab, goes into the nostril. This then goes into the cartridge. Then a little button clip closes it up. Everything's then sealed, completely sealed. Once it's sealed, you've contained the virus. This then goes into our nudge box where the actual cartridge will fit into the box. You close the cartridge, you close the box, and within just over an hour, the results will come through on your phone.
For 8 months now, we've been talking about viruses, viral mutations, DNA and RNA. But how much of that do we really understand? We thought we'd go back to the real basics and ask, what is a virus? Nucleic acids are large molecular structures known as biopolymers that form the essential components of life. They are split into 2 types, deoxyribonucleic acid, DNA, and ribonucleic acid, RNA. Every living thing on our planet is DNA-based, with the exception of viruses, which can be RNA-based. And some argue that viruses aren't living at all. So what are the roles of DNA and RNA? How are they different, and how do they interact? Both can be broken down into smaller sections called nucleotides, consisting of a phosphate, a sugar, and a base. The sugar separates the type of nucleic acid, being either deoxyribose or ribose. In DNA, the base can be one of 4 different types: adenine, thymine, cytosine, and guanine, which codes the genetic information, a bit like a 4-letter alphabet. In RNA, thymine is replaced by uracil. Each base has a singular opposing base to which it can bond, creating what is known as a base pair. Adenine always pairs with thymine or uracil, and cytosine always pairs with guanine. In DNA, base pairs form 2 complete opposing strands in a twisted ladder known as a double helix. RNA typically exists as a single strand, sometimes bonding with itself to create more stable structures. A living organism's entire DNA is called its genome. For humans, this consists of 3.2 billion base pairs. A person's entire genome is encoded in every cell of their body. It's split across 23 pairs of chromosomes, which exist in the nucleus of the cell. Sections of the DNA code for specific traits such as height, eye colour, and blood type. These coding sections are called genes. They're switched on or off depending on the type of cell. If they're switched on, they inform the cell's ribosomes to produce proteins which help provide that specific trait or function. This is achieved with an RNA strand called messenger RNA. Messenger RNA is the crucial link in understanding how viruses hijack cells, replicate, and then transmit. So what are viruses? Viruses are simple structures consisting of a small viral genome of either DNA or RNA protected by a structural protein. They're also very old. Computational biology has shown that a retrovirus existed 450 million years ago in the Ordovician period, where life existed only in the oceans with mollusks and early forms of fish. There's also a widely accepted theory that RNA viruses predate DNA-based living organisms, known as the RNA world hypothesis. All living things can be infected by viruses. They hijack cells, insert their genetic payload, and try to replicate. In some cases, this can permanently alter the DNA of the species they have infected. A study of the human genome found that approximately 8% is LTR retrotransposons. These are the remnants of viruses in our DNA that were passed down to us before we were Homo sapiens. Although some can be beneficial, viruses are often spoken about and understood as pathogenic or disease-causing. Most pathogenic viruses that emerge to attack human cells are RNA-based. This includes Ebola, HIV, influenza, and coronavirus. The average RNA virus is 11,000 bases in comparison to the 3.2 billion in human DNA. But they can cause huge amounts of damage. The reason RNA viruses cannot get much larger is because the enzyme that transcribes RNA for replication, called RNA polymerase, makes a huge amount of mistakes that never get corrected. So larger complex RNA viruses mutate too much to survive, whilst smaller RNA viruses are constantly mutating. RNA polymerase transcribes viral RNA to produce messenger RNA. This takes us back into the cell and where RNA viruses hijack DNA processes. The new messenger RNA from the virus goes to the ribosome, instructing it to produce replications. This repeats with more ribosomes being hijacked until the cell is full of virus replications, causing it to burst and die, releasing the virus to infect more cells. DNA and RNA have a very close symbiotic relationship, which can be incredibly deadly in a very specific set of circumstances. As with life, what is fascinating is how immensely unlikely those circumstances are, that an RNA virus, which has no intentions or motive, can mutate to just the right genetic formula to enter a human host, alter its DNA, replicate, and then transmit to infect another human host. And for that to happen at just the right moment. But it's a case of probability. The chances of this happening and a new virus emerging is increased as humans continue to push back the natural world, populating every corner of the Earth and moving more freely with air travel. They are both increasing their exposure to new viruses from wild animals and the risk of transmission between other humans. Without proper planning and containment strategies, including buffer zones between wild and domestic animals, the chances of a new virus emerging and taking hold in human populations, potentially causing the next deadly pandemic, is a case of when, not if. That's it for this COVID-19 special. Look after yourself and each other, and we'll see you again next week.